to improve euploid rates. All right, Tex, take it away.
Great. Thank you very much. Thank you, too, for Irvine Scientific for giving me the opportunity to be here today. And thank you all for coming. So we're going to start the presentation to talk about lactate, which is a critical component in our culture media, and how that affects stress and then ultimately numerical chromosome abnormalities. So by reducing lactate in our culture media, we have shown that that also reduces metabolic stresses to the embryos as they develop. And ultimately, we've been able to see that that also reduces numerical chromosomal abnormalities, so we're able to obtain more euploid embryos. So let me preface the talk by talking a little bit about some of the history. So Sir John Hammond, back in 1949, published a fantastic paper talking about how 8-cell mouse embryos were able to grow to the blastocyst stage. And this was really defined as the first true growth solution and basic easy components of physiological saline supplemented with some protein. So a few years later, 1957, Dr. Witten, as we are aware of, developed Witten's medium, and he was able to show that, again, by using pyruvate and lactate, we could overcome the 2-cell block in the mouse embryo, and that the embryos utilized this 3-carbon energy substrates more so than the 6-carbon glucose. So I'll get into that a little bit later as well. So murine embryo metabolism, also known as the mouse system. As we're all aware, 2-cell to 4-cell stage, the embryo really requires lactate and pyruvate. And then after about the 4-cell stage, going into the 8-cell stage, pyruvate uptake starts to decline in embryo culture and embryo development, and glucose metabolism sort of increases. And it continues to do so until it's the main substrate at the blastocyst stage in order to maintain embryo development and the main source of nutrients. Now, Dr. Gardner and Dr. Lane, back in 2000, really wanted to focus on pyruvate uptake as well as lactate concentration in the culture media. So as you see here, as lactate increases, pyruvate uptake also decreases. So really this big drop-off in significance is between 0 and 5 millimolar of lactate. Now, most culture media that's available commercially is around 6 millimolar. And that's an important note to keep in mind as we continue with the talk. They further showed that lactate in the media not only altered pyruvate uptake, but it also altered the metabolic fate of pyruvate. Now, getting into some of the systems of biology, microbiology, as we know, oxidation occurs in which lactate converts to pyruvate and vice versa. And with that reduction system, NAD is also produced and NADH. So under normal circumstances, this reaction with LDH, lactate dehydrogenase, it catalyzes to very, very close to equilibrium. So normal systems, lactate and pyruvate basically balance each other out. But if we have high concentrations of lactate, LDH exhibits sort of a negative feedback inhibition, whereby the rate of pyruvate conversion of pyruvate to lactate is greatly decreased. And that ultimately has a profound effect on the decrease of NAD production and regeneration. So, of course, NAD is very much required for glycolysis and to convert glucose into energy. So hopefully I haven't lost you yet. As we're all pretty much familiar with biological chemical pathways, let me kind of make it a little bit easier for you here. So you have high concentration of lactate. That then has a negative feedback on LDH, which catalyzes pyruvate into lactate and vice versa. Pyruvate does not convert to lactate with high concentrations. Decrease the NAD-NADH ratio. Therefore, that affects the conversion of glucose to pyruvate through glycolysis and ultimately impacts energy conversion pathway in the long run. So Dean Morbick also published a paper relatively recently looking at all the different compositions in culture media. And he was able to obviously show as well the different concentrations of lactate in different culture media. So going back to my original slide where it talked about 6 millimolar, what Irvine Scientific has done is decrease that millimolar concentration of lactate from 6 to 1. Now, I had two different laboratories make the transition to a low lactate culture media. Laboratory 1, or Laboratory A, predominantly used antagonist cycles, Lupron triggers. They previously went from a sequential culture protocol to the CSCM in next generation in the beginning of, 2017, December. We culture in an environment of about 6688. And they do undisturbed culture from day 1 all the way through day 5. They do not look at the embryos on day 5, or day 3, excuse me. They culture all the way from day 1 to day 5, day 6, and day 7. And they use a no renewal system in which they do not renew the culture media on day 3. Same culture media drop all the way for 7 days. Lab B, fairly similar, antagonist cycles predominantly, HCG triggers. We previously used CSCM, undisturbed culture media, but we made the transition to the low lactate media at the beginning of this year. Roughly about a 88% culture environment, and undisturbed culture on day 1. We do look at embryos on day 3, where we do some assisted hatching for PGTA, followed by day 5, day 6, and day 7. Again, a no renewal system. So as we all know, blast utilization rate is a predominant KPI for our laboratory. It really gives us an understanding of how our embryos are growing and progressing with our culture environment. So when we looked at Lab A and compared CSCM NX, which is the low lactate, versus a sequential system, we really didn't see a significant difference in the blast utilization rate. Pretty much things were exactly the same. When we also looked at the Lab B, which went from a CSCM high lactate to a low lactate culture media, we pretty much saw the same thing, not a significant difference in blast utilization rate. However, other laboratories, Jason Barrett in Los Angeles and Beverly Hills, California, he was able to show that through time-lapse and using the mouse system, the embryos that were available for biopsy seemed to be a bit faster and more ready for the actual procedure when embryos were cultured in the low lactate media. And he was able to show this through time-lapse. Now looking at in the human system, he was able to see a significant difference in good or fair quality blastocysts on day 5. So about a 5% difference, 5% more embryos in the CSCM NX versus the CSCM. So more interestingly, in our laboratories, we then wanted to do and look at ploidy rates. And we do a lot of PGTA. One laboratory, Lab A, does about 60%, whereby the Laboratory B does about 70% of all cycles. So the percent euploidy rate in embryos, now this is defined as a 30%, less than 30% gain or loss of particular chromosomes. When we looked at about 3,000 embryos, we saw a significance of about 5% more euploid, chromosomally normal embryos identified by using a low lactate media versus a sequential system. Now looking at Lab B, again about 4,500 embryos, we saw a greater significance of about roughly 10% more euploid embryos cultured in CSC NX versus CSCM. So this is quite significant for us, especially if we're able to obtain more euploid embryos. And for those who do not do PGTA analysis, this is an added bonus of simply switching a culture media. Now we'll keep in mind and let you know that, again, the only difference made was a change in culture media. Our culture system was exactly the same. Stimulation protocols were very, very similar between the two systems. So by simply changing out the culture media, we did see a difference. Now looking at implantation rates, again, between sequential and low lactate, we did see a significance in Lab A with implantation potential from embryos grown in the low lactate. And we saw similar but not significant, but the ongoing trend in Lab B for implantation potential. So another lab in Turkey has pretty much published and poster presented here at ESHRE, where again their KPIs did not necessarily show a significant difference in fertilization rate, embryos, slow or fast embryos on day 3, or blast development, but they do see a positive trend similar to what we've seen in clinical ongoing and implantation rate improvement with the low lactate culture media. So again, drug stimulation routine did not change, culture environment did not change, stimulation drugs were very, very similar. The biopsy technique, which we take very seriously, did not change. The PGTA platform, all done on Illumina MySync with next-gene sequencing, did not change. In fact, the laboratory in which we did the embryo, the laboratory in which we did the embryo biopsies, but of course the genetics laboratory is all in-house. we did mix with different outsourcing of genetic companies, as well as our calling policy did not change. The only thing that changed was the low lactate in the culture media. So let's talk a little bit about types of chromosomal anomalies. As we're aware, we have numerical as well as structural. The most familiar, the aneuploidies, the monosomy, and the trisomies. So under normal mitosis, as we're all aware, we've distinguished and be able to carry over the correct number of chromosomes, but anaphase lag is where sister chromatids is lost due to improper spindle formation. That's where we see a lot of our monosomy effects, as well as sister chromatids and non-disjunction is where we predominantly see triploidy embryos. Now, we do know through literature that these do have an effect on embryonic stress. Embryos that are stressed do have more incidence of anaphase lag, as well as non-disjunction and, of course, errors in cytokinesis. So the idea there is that by introducing a less stressful culture environment, you're going to have more normal chromosomal segregation. So as we're aware as well, aneuploidy, most frequent cause in chromosome missegregation during meiosis and mitosis. During this case, we're really focusing on the mitosis events, or mitotic events. Temperature changes, oxygen concentration, culture media, and hormonal stim regimes could affect spindle assembly and therefore chromosomal segregation. Aneuploidy alters dosage of genes on affected chromosomes, as we're all aware, and aneuploidy itself can introduce certain cellular stresses, which in turn can create additional chromosomal instability. A great paper that just came out, Cellular Stress Associated with Aneuploidy in Cells, 2018, really talks about and highlights the effects of stress, and particularly mitotic stress with these lagging chromosomes, which then have a knock-on effect with replication stress and slowing of the replication fork and progression, followed by, again, the increase in ROS levels in the mitochondria. So in general, aneuploid cells themselves produce more ROS, which and then increases damage to the DNA and leading to replication and mitotic error. So again, sort of a vicious cycle with some of these aneuploid cells. Now again, just to recap, lowering concentration has been shown to reduce metabolic and mitotic stress, and ultimately in our laboratories we've been able to identify that this hindrance of stress to the embryo has caused an increase in chromosomally normal embryos. And with that, I recommend you de-stress, enjoy Barcelona, and thank you for listening.
Thank you, Tex. We're going to take questions at the end.
Perfect.
So you can have a seat, and it's now my great pleasure to introduce our